Hello everyone, this is my new video and here we're going to talk about the B cell function. In the first part, we will focus on the main duty of B cells, creating a big diversity of immunoglobulins. First of all, a quick recap. In a previous video, we saw that all cells in our blood come from the multipotent hematopoietic stem cell in the bone marrow. They first become myeloid progenitors or lymphoid progenitors. Lymphoid progenitors will differentiate and become either natural killer cells or T lymphocytes or B lymphocytes. We remember these three types of cells that come from the lymphoid line from the first letter NTB. NTB means not in the body because these three types of cells kill everything that does not belong to our body. Easy to remember. Now, NK cells are killer-trained soldiers. They kill everyone with no distinction, no specific memory. The T cells are the generals. They help each other. They organize the others and they tell them what to do. B cells are sophisticated IT engineers. They produce very specific weapons against the enemy. These weapons are called antibodies. So now that we made this quick recap, let's see what the B cells, the B lymphocytes, are made for. A B cell's main mission in life is to produce a vast diversity of immunoglobulins, a diversity of antibodies. As humans, we have to deal every day with numerous different pathogens, such as viruses and bacteria, which can also be mutated or resistant. So the key to fight all these intruders is the diversity in the production of our antibodies. How the B cells can assess this diversity while producing antibodies? First of all, let's make one thing very clear. Antibodies are immunoglobulins. It's the same thing. Before that, they were also called gamma globulins, but not anymore. So antibodies and immunoglobulins, it's the same thing. Antibodies are nothing but proteinic receptors on the surface of the B cells, allowing them to connect to different antigens. So immunoglobulins are antibodies, and they are also proteins, and they function as B cell receptors, called BCR for short. The structure of the antibody forms the letter Y. Antibodies are made of two heavy chains, the big ones in green-blue color, and two light chains, the small ones, in pink, orange, red color. Every heavy chain and every light chain have a region composed of amino acids in a stable order, so this region is called the constant region. Every heavy and every light chain contain also a region with amino acids that change their order all the time, and so this is called the variable region. This is the most exciting one because the unique order of amino acids for a specific antibody makes it capable of binding to a very specific antigen. Let's take a closer look and see how does this happen. Now imagine small squares like genes in the part of the DNA of the B cell dedicated to define the immunoglobulin structure. The green genes code for the constant region of the heavy chain, so they will not be modified. They will remain the same through all the stages of B development. The gene segments that code for the variable region of the antibody will be recombined to create the vast diversity of this region. This whole process is called VDJ recombination. Remember, these genes are variable, they are also diverse, and they are joining each other to form different groups, providing our body the extraordinary function of producing diverse antibodies. First, the genes of the DJ section are combined, and they form the DJ gene section. And then, a part of the genes of the V section is added, forming a section called VDJ. So when this part of genes is transcripted to mRNA and then translated to a protein, we get the variable portion of the antibody. Any of these small squares can be combined to the others in a different order, and it is the random combination of these segments that creates the vast diversity of antibodies. Fascinating, right? Okay, so when the B cells start creating this immunoglobulin diversity, well, gene segments of the variable region of antibodies, in order to create diversity, starts very early. It starts even at the stage of the bone marrow. Remember, in the bone marrow we have very young B cells at the stage of the pro-B cell and at the stage of the pre-B cell. They start already creating immunoglobulins with constant and variable regions by recombining the genes of the variable section, so they create this diversity. It's exactly the way we explained before. So, at this stage, the lymphocyte leaves the bone marrow to start the lonely journey until meeting the antigen. The B cell is called now a naive B lymphocyte. At this stage, the naive B lymphocyte has only two types of immunoglobulins, IgM, IgD. We will explain in details the function of each type on upcoming videos. So the lonely travel starts. Naive B cells constantly travel in the blood between the spleen and the lymph nodes again and again until they will meet an antigen. But why this travel is lonely? Because meeting the antigen may never happen. The poor little lymphocytes. There are three scenarios for them. 1. The B lymphocyte meets nobody, so the lymphocyte dies with apoptosis. This is a very lonely existence. 2. The B lymphocyte meets the wrong antigen. B cells think having met the correct antigen, but in reality it is just a self-antigen. Our body is capable of eliminating the falsely activated B cells or to force them to die, to fall in anergy. And this process is important because it protects our body from autoimmune diseases. The third case scenario is that the lucky B lymphocyte, the one that has the unique combination of amino acids, and makes the exact perfect match of an antigen. So, once the antigen matches the antibody, the B lymphocyte becomes activated. Now, the B lymphocyte can go to the germinal center for further maturation and specialization. Imagine the germinal center as a kind of university where the B cells take their special training. They become experts in recognizing very specific types of antigens and produce 3 other types of immunoglobulins: IgA, IgG, IgE. So after antigen activation, the naive B cell that produces only IgM and IgG will go from school to university. And now they will learn to produce IgA, IgG, IgE immunoglobulins, and this process, since they have changed class, is called class switching. Great. So that was the end of the part 1. Next, we're going to see what happens in the germinal center of the B cell and a way to remember it easily. Thank you for watching.
Explore our extensive library of over 1,800 video lectures to learn about a wide range of topics. Only on sqadia.com.